Hello, welcome to this journal club. I'm Xiaoxu Yang from Joseph Gleason's lab in the University of California, San Diego, and Rady Children's Institute for Genomic Medicine. I'll be presenting our work together with my former colleague, Dr. Martin Boyce, now an assistant professor in the University of Colorado School of Medicine. The work we are talking about today is titled Developmental and Temporal Characteristics of Clonal Sperm Mosaicism. Before going into our topic, we want to lay the foundation about the problem we are studying. Assuming you have a couple that has normal behavior and doesn't have any disease or certain phenotype, but when they give birth to several child, one of them was diagnosed with certain phenotype or with some disease. What would they like to know is the probability if they have another child and the child has exactly the same disease or the same phenotype. This is called the recurrence risk in this family. As we are a genetics lab mainly focusing on recessive disease in the past, we know for sure that if both parents are the carrier of the disease allele or phenotype-related allele, the probability of recurrence is a fixed 25%. However, for those de novo mutations, which by definition you can't see the risk allele or variant in any of the parents, at least for the blood, you do see the disease-causing variants in the child with the phenotype or the disease. So what's the probability to see a recurrence is actually unknown. From the population level, it is estimated that there is around 1% recurrence risk, but whether those 1% recurrence risk actually comes from 10% families that each have 10% risk, or 1% of families in the population that has 100% risk, or every family has an average 1% risk. It's completely not known. And for the de novo mutations, our previous study suggested that there might be some mosaic mutations from the father or the mother that are not detectable from their blood, but actually transmitted down to the child and then cause the recurrence. To compare those individual precise risk with the imprecise population risk, we have to lay the foundation of normal mosaicism in the population. As we know from previous literatures, that almost 80% of those de novo mutations come from the paternal haplotype. What we want to study is whether those mutations can already be detectable in the father's sperm as mosaicism. Our major research questions for this study. First, what is the normal mosaicism observed in sperm? Second, what is the inter-subject and intra-subject variability? And as people have previously reported, there seems to be a paternal age effect of those de novo mutations, meaning that older males will transmit more mutations to the child as de novo. So we also want to know about the differences between those clonal mutations from old and young individuals. And third, how can we use mosaicism to learn about the sperm lineage and also compare with the other progenitor lineages from the same individual? To answer all these questions, especially for the inter and intra-subject variability and the effect of age on sperm mosaicism, we designed this control cohort. We first collected 12 individual donors we define as young age, between the age of 18 and 22. 3 sperm ejaculates were collected between the 6 and 12 months. We also recruited 5 old men at the age of more than 48, and we defined them as advanced age, and we also collected sperm ejaculates as well as their blood sample. We further subject all those samples to 300x whole genome sequencing. We contrast the sperm sample and the blood sample to see blood-specific sperm and blood sharing and sperm-specific variants, which are mosaicism. And now Martin will tell you more about the inter-subject differences.
We first assessed the inter-subject variability, which you can see here. We've plotted the number of variants for sperm-specific, shared, and blood-specific variants that are clonal mosaic. A typical individual has a higher number of sperm-specific variants than shared variants, but the highest number of clonal mosaic variants in blood. On average, a YA cohort individual harbors around 30 clonal mosaic variants in sperm, which are the sperm-specific and the shared variants together. We next wondered what the allelic fraction of these variants would be. So here you can see a plot of the square root transformed allelic fraction where you can appreciate that the tissue-specific variants in sperm and in blood actually exhibit a lower maximum abundance, or also average abundance, than the shared variants. We can look at this in a different way when plotting the ranked plot of all the sperm mosaic variants, both sperm-specific and shared. And you can see again that they are kept at a lower allelic fraction for the tissue-specific variants. And overall, there's also a difference in the shape of the curve. This together suggests, first of all, that the shared variants indeed originate earlier in development and therefore are more widely distributed at higher allelic fractions, but also that the accumulation of these mutations is distinct between the sperm-specific, tissue-specific variants and the shared mosaic variants. We next assess the intra-subject variability, which means the variability of clonal mosaic variants in sperm from one sample to the next within one individual. For this, for several of the YA cohort individuals, we collected multiple sperm samples to track the clonal mosaic variation across time. We used this in a time frame of 6 months and 12 months for the second and the third sample. And we used a method called targeted amplicon sequencing to allow for highly sensitive detection of clonal mosaic variants and a very high accuracy of the allelic fraction. So when we did this for the 3 sperm samples and a selection of clonal mosaic variants, we actually found that within the different time points, you can see here, for instance, time point 1 and time point 2 of the sperm allelic fraction in a square root transformed visualization, we have very high correlation between the different time points. We also find that there are actually no individual variants that drop in and out of samples, which means that they are stable within this time frame of 1 year. You can see here a plot of the absolute change between the different time points, while the maximum allelic fraction change is around 0.02, 0.03. We do find that most of them exhibit very little variation, suggesting that clonal mosaic variants in sperm are stable within an individual.
Based on this stable intersubject variability, we actually can look at the intrasubject variability and compare the age effect between the number of sperm mosaicism. When we compare the number of mutations found in each of our young donors and the advanced age donors, and show here, as the age on the x-axis grows, you can see a significant growth of number of detectable variants, as shown in y-axis, from the blood, shown as orange triangles, whereas the number of mosaic variants that are shared between the blood and sperm, or that are specific in sperm, shown as the green circles, are very stable across decades of years. This is partially because of the different clonality of the sperm and blood progenitors when men age. We further combined another deep sperm sequencing cohort we previously estimated, which is defined as rich cohort, as we see a stable number of sperm-specific shared and young blood variants. We split out the old blood variants, and then from the 4 categories you can see there's an exceeding number of the variants detected from the old cohort. By analyzing those 25 individuals, we look at the base substitution signatures and found a dramatic decrease of T2C mutations, which are age-related, compared with a 95% simulation confidence interval from the de novo mutations found in the Simons Foundation, which means that most of the variants we observe are actually early embryonic stage. We further estimated the number of mosaic variants from all the 4 different categories, and there are on average 30 sperm detectable, which include both the green sperm-specific variants and the brown shared variants from each individual, and is stable over time. And by looking at the exonic variants, as we have around 1 per individual on average. After looking at the functions of those variants, we estimate that there is around 0.1 with CAS score larger than 25 or loss of function, which is predicted to be functional if the variant was actually transmitted to the offspring. We further calculate in 100 individuals, there are around 28 loss of function or CAS score larger than 25 which we define as C-log variants, and around 7.2% is haploinsufficient. And among them, 1.3% actually are defined by the Simons Foundation as an autism risk gene. So in summary, 7.2% main harbors a likely pathogenic clonal mosaic variants in the population. And also, the deleterious mosaic variant is stable across age and contribute to around 15% autism diagnosis resulting from de novo mutations previously estimated, and also a similar percentage in congenital heart disorder.
What is the consequence of clonal mosaic variants on de novo mutations that are transmitted to offspring? We have not touched upon the non-clonal mutations, but we have described clonal mosaic mutations that originate in the development of the father. Collectively, these clonal mosaic mutations, together with the non-clonal mosaic mutations, will actually be transmitted to a fetus and contribute to the paternal de novo mutation landscape. So what has been described before is that paternally contributed de novo mutations increase with the age of the father. Based on our results, we believe that this is not due to the clonal mosaic mutations that appear to be stable with paternal age, but only due to non-clonal mutations. Consequently, the relative contribution of clonal mosaicism to the de novo burden decreases at father's age. The father's age is also important when considering the false positive and the false negative rate when looking at blood for mosaic mutations that may be transmitted to an offspring. You can see here that the false positive rate actually will increase with the age of the father, and this is due to the clonal mosaicism expansion in blood. The false negative rate, however, will stay stable, like the sperm mosaicism does. And you can see here why it is important to look for sperm mosaicism if you want to have an accurate picture of the mosaic variants that may be transmitted to offspring. In summary, we have demonstrated that if you look at young age cohort and advanced age cohort, that the number of clonal mosaic variants in blood increases with age, in some individuals by an order of magnitude or more, and the allelic fraction changes dynamically consistent with an increase of clonal diversity. In contrast, sperm clonal mosaic variants do not change with age, neither in their number nor in their allelic fraction. We believe that this is an important insight, allowing us to use sperm mosaic analysis to determine the lifelong threat of transmission of mosaic variants to offspring. We also found, based on the calculations from our cohort, that we predict that 1 in 15 males actually harbors a mosaic mutation in around 5% of their sperm on average, that is actually likely incompatible with life or can lead to congenital disease.
We would like to thank Joe for all the supervision, and thank Xin, Danny, and Renee for the bioinformatic help. We thank Valentina and Kelly for the clinical coordinating. We thank Jennifer and Laura for all the help in human genetics. We also want to thank our collaborators, Jonathan Seibert and his lab, and all the participating individuals for their donation. We would like to thank Rady Children's Institute of Genomic Medicine, UC San Diego, Simons Foundation, and NIH for the funding support.